Hi there, thanks for joining us for another edition of Biology Notes. Today we'll be examining heredity and Mendelian genetics. Oftentimes we're curious about why organisms look the way they look and why they work the way they work, and we're going to be exploring how traits are passed from generation to generation. So to start off, we're going to look at a couple of vocabulary terms. The first is heredity, and this is simply the passing of characteristics from parents to their offspring. And the branch of biology that focuses on heredity is called genetics. And this will be our focus of study for the next couple of weeks. Gregor Mendel truly paved the way for our modern understanding of genetics. We consider him the father of genetics. He was a monk, and at his monastery he carried out a number of experiments on pea plants. He crossed different pea plants and then observed the traits of the offspring, and this better helped him understand how traits are inherited. It might seem a little bit strange that this monk was fascinated by peas, but it turned out to be a huge advantage that he worked with this organism. There were several reasons that peas were such a good model to study. First of all, pea plants have male and female reproductive parts enclosed on the same structures, and mating can easily be controlled. Pollen can easily be transferred from one plant to another, and there aren't any ethical implications about the mating of plants. A second big benefit to plants is that they're small and easy to grow. They also mature very quickly. We don't have to wait 20 years for them to grow up to see what they're going to look like. And peas have many offspring. That means we can get results very quickly, and there are plenty of subjects to count. Finally, peas have traits that exist in very clear forms. There aren't any in-betweens. For example, pea plants can be either smooth or wrinkled. While organisms like humans have a lot of in-betweens, we're not typically just tall or just short. There are a lot of options in the middle. Pea plants are really an either-or. Not just the idea of having round or wrinkled seeds, but their seeds can also be either yellow or green. Their flowers can be either white or purple, and so on. There aren't any intermediates, which makes it a lot more simple to analyze results. There's several key ideas that form the foundation of genetics, based on what Mendel saw and based on what we've learned since his time. First of all, for every inherited characteristic that you have two copies of instructions. One of those copies came from your mother, and one of those copies came from your father. There are different versions of genes. There are different options for the codes that you can inherit, and these are called alleles. For example, when we look back at flower color in pea plants, we know that there can be a purple version or allele or a white version or allele. And we can see this illustrated in this diagram. We can also see that when we look at chromosomes that code for the same inherited characteristics, we can call these homologs or homologous chromosomes. Every diploid cell, or your somatic cells, have two sets of homologous chromosomes. You get one set of instructions from your mom and one set of instructions from your dad. So again, pictured here is a pair of homologous chromosomes, showing us that they do have similar traits. They're both coding for flower color. One just happens to have a purple allele, and one just happens to have a white allele. In the simplest form of inheritance, when two different alleles are present, for example, a version of the purple flower color and a version of the white, one of these versions might be completely expressed, while the other one might not have an observable effect. Meaning that the expressed form, the form that we actually see, is what we call the dominant trait. And we look at this as a capital letter to show that it is dominant. It is the version that will be seen. On the other hand, we use a lowercase letter to show a recessive trait, meaning the trait that's not expressed, the version that you don't see because it's hidden by the other. Now, when gametes are formed, the alleles for each trait separate. So even though that pea plant has a white copy and a purple copy, they only get to pass on one of those copies. The gametes only carry one allele for each characteristic. This works because during fertilization, gametes unite, the sperm and the egg come together, and each gamete then contributes one allele to the then diploid offspring. This idea that we only pass on half of our instructions leads us to a quick review. During which process are our gametes formed? That's right, gametes are formed through meiosis. So in meiosis 1, what actually happens to our homologous chromosomes? Good, they separate. Our homologs separate in meiosis 1, and then the sister chromatids are separated in meiosis 2. Finally then, how many alleles for one trait does each parent get to contribute? The answer to that is only one. We only pass on half of our instructions to our kids. There are a lot of important vocabulary terms that go with this section. The first is the genotype. And the genotype is the genetic makeup of an organism. This is saying which genes did the individual get, which set of alleles do they have. So typically when we describe a genotype, we use letters. We'd say this individual is big B, big B. This individual has big B, little b. This is describing the versions of the genes they got. The phenotype is the physical appearance. So for example, if we had a genotype of big P for a pea plant, their physical appearance would be purple. Now we also have two terms that can tell us if the letters within the genotype are the same as each other or if they're different. So the prefix homo tells us same, and if an individual is homozygous, the two alleles that they possess are the same. So they can be either homozygous dominant, big A, or they could be homozygous recessive. Two of the same, recessive though, little a. The opposite of homozygous is heterozygous, and that prefix hetero tells us different. And this means that the individual has two different versions, or two different alleles present, being big A and little a. As we look at the diagram at the top, this reminds us that we are looking at homologous chromosomes. And as I look at a copy that came from mom and a copy that came from dad, this individual is homozygous dominant because they inherited a big B from each parent. The heterozygous individual, however, got one of the homologous chromosomes in the dominant version and the other in the recessive version. These are different size letters, different versions, therefore this is a heterozygote. When we look at inheritance or passing on of traits, it's easy to see that if an individual plant passes on a purple allele or a dominant version of the gene, and if the other parent also passes on a purple allele, the offspring would result as homozygous dominant, big A, and their phenotype would be purple. We can see if an individual plant passes on a purple allele and the other parent passes on a white allele, that resulting offspring will have one of each, so they'll be heterozygous, big A, little a. Because the dominant gene is purple, this individual's phenotype will be purple. It takes on the appearance of the expressed purple dominant trait. The only way for a plant to end up as a white pea plant is if it gets a white allele from one parent, a white allele from a second parent, little a, and we would call this individual homozygous recessive, and their phenotype would be white since they do not have a purple version. Oftentimes in our study of heredity, we like to make predictions about potential offspring from mating events. And we use what's called probability or a likelihood that a specific event will occur. Mathematically, probability equals the number of times the possible outcome can happen over the total number of possible outcomes. Well, that's a little strange sounding. Let's keep it simple and think about a coin. What is the likelihood or probability of tossing a coin and having it come up heads? Well, how many heads are there on the coin? Just one. But how many total sides are there on the coin? 2. So that means the probability for tossing a coin with heads would be 1 out of 2. And we can also express that as a percentage and say 50% of the time you are likely to toss a coin with heads. We can also ask ourselves about the probability of a couple having a child that is male. And the answer every time a couple has a child is 1 out of 2 or 50%. There are 2 different genders possible, and one of those is male. Now, we can take this a little bit further and ask ourselves about the probability of a couple having 4 boys. Having a boy carries the likelihood of 1/2, and in order to find out the likelihood of multiple events all occurring together, we're going to multiply. So the likelihood of the first boy being a boy is 1/2. The second child also has that 1 out of 2 probability. Every time gametes are formed, there's a 50-50 chance of which genes will show up. And so we take that 1 out of 2 probability for each of those 4 boys, and as we multiply out 1/2 times 1/2 times 1/2 times 1/2, that gives us 1/16. We're going to start our predictions of probability and inheritance with monohybrid crosses. The prefix mono tells us 1, and a monohybrid cross is only examining the passage of one trait from a generation to another. So here we're going to be examining one trait, and that is fur color in rabbits. And here they're telling us that the dominant allele is for white fur, and the recessive allele codes for brown fur. So we're going to set up a device called a Punnett square, which will allow us to predict what the offspring of this cross may look like. Please continue on the second portion of the heredity and genetics video to see a few practice problems. Thanks for joining us.